Good afternoon, everyone. I am Terry Young. I serve as the Biochemistry Molecular Biology Section Trustee. I am deeply honored to join you today to provide the history of the Friedenwald Award. This award was established in 1957 as a commemoration to Dr. Jonas S. Friedenwald, a distinguished researcher whose contributions encompass the entire field of ophthalmic investigations. This award is presented to an individual annually in recognition of outstanding research in the basic or clinical sciences as applied to ophthalmology and the visual sciences. The 2024 Friedenwald Award is presented to Anand Swaroop, PhD, who's a senior investigator and chief of the Neurobiology, Neurodegeneration Repair Laboratory at the National Eye Institute. Dr. Swaroop's research is focused on genomic regulation of retinal development, aging, neurodegeneration, and age-related macular degeneration. It is my pleasure and honor to present the Friedenwald Award to you, Dr. Swaroop. Please come forward. Now I am pleased to introduce Dr. Donald Zak, Professor of Ophthalmology at Johns Hopkins University School of Medicine. He will review Dr. Swaroop's achievements and introduce his lecture.
So I managed to get up here without falling down the steps. That was good. As everyone says, but in this case, it truly is such a tremendous personal honor, joy, everything else to tell you a little bit about Anand. Unfortunately, I won't tell you too much about his science, since he'll be doing that better than I can, but I'll tell you more about the person and how many people he's affected. But he's just been an amazing friend, colleague, and collaborator. So this is just a little warning that there's no such thing as privacy anymore, because everything gets captured on a cell phone. So this is about a year ago in New Orleans. We're having dinner with Anand and Serge Picot, and the cell phone goes off. And who do you think he's talking to? He's talking to Pat DeMore, who was just telling him that he was the 2024 honoree. So it's captured. You can see there's a little bit of shock there, but mostly joy. And he's obviously totally deserving. He's done so much in science. He's affected so many people. Started with transcription factors that you'll hear more about, and it's moved into complex networks of regulation, AMD, a basic scientist who's applied his basic science to really understanding patients and disease, and hopefully will lead to new treatments. This is a picture from a few years ago, back when Anand had more hair, and I had a little bit more color, but Anand reminds me, since he has better memory than I do, that we first connected in the early 1990s. And at the time, I was a postdoc in Jeremy Nathan's lab studying rhodopsin regulation, and Anand was studying NRL and transcription factors, and it was such an obvious marriage in science that he contacted me, and as I said, we've been friends, colleagues, everything since then. Many stories that I can't share with you today, but we'll do that over the bar, but many stories that I will share. So let me just give you a little factual background, then we'll get back to the fun stuff. Anand got a BS in chemistry, zoology, and botany from Beirut University, and I apologize if my pronunciation is not perfect. Then a master's in biochemistry from GB Pant University, and then at the very prestigious Indian Institute of Science, got his PhD. Then he did his postdoctoral studies at Yale, and as you can see, he worked with 3 of the real giants in human genetics. So he got incredible training, then left to become a faculty member at University of Michigan, where he progressed from assistant professor to associate, full professor, named professor, and then Paul Sieving made the brilliant insight to recruit Anand to NEI in 2007. And most of you probably already know this, but it's interesting that he was able to figure out a name for his division that fits with his favorite factor, NRL, neurobiology, neurodegeneration, and repair. So in addition to being a great scientist, Anand really takes care of the people in his group, and he really likes to train people, really likes to test them a little bit, but really has trained so many people. And this just shows one of the many parties that Anand has sponsored for his research group at ARVO in the lab. He's also a great cook, but I must say, having seen Emily's wedding cake today, this is Anand making cookies. So it's not quite a wedding cake, but I'm sure it was still very tasty. This is one of his parties at ARVO. ARVO always has an Anand party. This is his backyard. Many people remember parties, and some of us have been lucky enough to be invited there. And Manju always throws the best parties. And this is just going back another few years. So this is, I don't know the year, but it was early in Anand and Manju's marriage in both New York and New Haven. Is that right? Or Michigan? Not sure exactly where they were, but obviously there. You can see the young Anand there. He was wearing a tie then too, so this is the 2 times I get to see Anand with a tie. As important as his science and his colleagues are, Anand's greatest love, obviously, is his family. And this shows Kanchan and Alok, but they were in their single digits, and they're here today to share the wonderful day, and you can see how they've changed somewhat, but there's still similarities. And there is Anand, the happy grandfather, with Ruhi, and she's so cute, and some people got to see her last night at the party. This is Anand, who's quite a dancer. I don't know if you've all seen some of his clothes, and he's, again, dancing. A happy kind of guy. And for some of you who had the pleasure of this lovely party last night, this is hot off the press from the tower, seeing the wonderful views. And lastly, we're almost at the important part, which is Anand's talk. I've been trying to collect stories about Anand, and I'm about to share some quotes from people who I promised their identity is preserved. You can try to guess, but just like study section, when you try to guess who the reviewer was, you're wrong more than half the time. So, and actually, coming over here today, there was another colleague who Anand knows very well, who gave me an additional story about Anand, which I didn't have time to get in here, but that quote was something to the effect, Anand has so many brilliant ideas, he himself can't even keep track of them. But anyway. All right, so here are the observations from colleagues, friends, and family. Anand tells it the way it is. And for those who know Anand, that's one of his strengths. I mean, he tells you what he thinks, and yeah, so be it. His parents used to tell their kids to behave, or if they didn't behave, they'd get a lecture from Anand. But don't worry. They always learn something new each time. In my interactions, I always learn a lot. His unsolicited advice is always too long and meandering. Maybe I'm doing that a little too. But it inevitably contains exactly what you need to hear, whether or not you want to hear it. I think teaching is one of Dad's callings. I guess the dad might give a little hint. I think teaching is one of Dad's callings in life, maybe his main one. He loved to teach us as kids, and even now, he tells everyone the story of how, on a road trip to Philadelphia, where he explained the inheritance of red-green colorblindness, which we're both actually red-green colorblind, is what first sparked his son's interest in genetics and biology in general. And lastly, for today, more at the bar, he loves to brag about the successes of his students and his postdocs and his kids, no matter how long ago they were in his lab. And lastly, and most importantly, everybody agrees Anand is the best. And we're so happy for him, for his presentation. And now I want to introduce Dr. Anand Swaroop. Congratulations.
But what can I say? First of all, thanks to ARVO. But before I do that, I'm delighted to be here, particularly sharing this stage with Emily, my good friend and colleague from NEI. I'm humbled, I'm delighted, I'm honored. What else I can say? I think it's great to be here on this stage, particularly for someone who never came first in his class. And the fact is that I've told this story to many, that both where I grew up, my native country, as well as this country, has been amazing to me. I came to this country with $23 in my pocket, and look where I am. So if I can do it, you can do it. So let's start with that. First, let me thank ARVO. Obviously, I've been coming here since 1988. That was my first ARVO, and I think I never missed any. The awards committee, selection committee, the nominators, Don, Emily, Eyal, and David Gam. Thank you all very much. You saw a couple of those pictures. The picture in the center particularly is another one from the same dinner that we were all together. This was the next day party that we had, and this was back in the lab. So thank you again to ARVO, and I'm truly honored. Now, the title of my talk is Genes, Networks, and Karma, right? And genes and proteins, they don't function alone. They have to be part of these multi-protein complexes within the cell. There's a whole network. Without that network, they're useless. They will not do what they're supposed to do. Same thing is true for me. I am blessed to have an amazing, amazing group of friends, family, colleagues, collaborators, mentors, teachers, and my journey has been nothing but joyful. Now, so what I decided to do was, rather than tell you all about my science, I thought I'm going to acknowledge the people to whom I will be always grateful. The people who made this journey possible and made this journey joyful. So I'm going to first tell you the people who are here, and I'm not going to talk about their work. Soumitra Paul, who is going to be talking about evolution of nocturnality. Kiam Preston, about NRL interactors. Sophia will be talking about afrin A10 receptor, which is highly expressed in cones. Zip and Q, about NRL-centered gene regulatory networks. Mohita could not be here, but Matthew Brooks will talk about her work on histone lactylation. Zulong Liang will tell you something more about NRL that you probably don't know, but I need to correct. I just remember, Don said something wrong, and that is NRL was not the name that I gave it to my lab. It was Sheldon who recruited me. So Sheldon, thank you very much. Zulong will tell you something new about NRL. I think it's rather provocative and different. Nivedita is going in her own new direction, and she will tell you about genome topology, intrabicular meshwork cells, going into glaucoma. Tingyi Lane, a graduate student, and Matthew Brooks, both will present their work on aging of the retina. And Anupam Mondal, who actually talked yesterday, and you heard about some of the work he had. He also helped me make some of the network slides and all. So Anupam is doing the network medicine, and Minji Kim is going to tell us about our attempts to do drug discovery. So I won't be discussing any of their works. Now, I want to tell you, I have to do this because I'm so grateful to my parents who would have been proud, and I'm sure they are. So I was born in a small village in India, and my parents were the first teachers. They are the ones who taught me honesty and tell what it is. The principles, never compromise on those principles. And integrity, those are the values they always told. My mother never saw any fault in me. She was the biggest supporter until she saw me drinking a glass of beer. And my wife will tell you how upset she was. She won't talk to me. And I was 33 at that time. So, and my grandma gave me the name I have, and my grandpa actually told me numbers and tables while counting the stars on a nice summer night in India. My wonderful sisters, my spiritual guru, and two of my oldest friends for almost 55, 60 years, who all gave me the directions, who helped me when I needed the most in my early days as teenager. And serendipity then took me to do masters in Pantnagar and graduate school in Bangalore. I don't know how I reached there, but I did. And I had amazing group of friends, my classmates, Yogendra Singh and bunch of other classmates here who became professors in their own rights, and my mentor, Professor Ram Sharma, who tried to keep me in mitochondria. And I loved it, but these two other gentlemen here, Professors Padmanabhan and Gopinathan, they ignited my interest in genes. And I loved genes and the recombinant DNA, Herbert Boyer and Stanley Cohen's early experiments with that, and then Maniatis gene cloning. That's what really took everything away from me. And nobody will give me a fellowship except Professor Alan Garan at Yale. And he taught me or introduced me to Drosophila genetics, and I cloned my first gene. One of the happiest sort of days in science for me when I cloned P150 glued. Uta Franke, I went to her lab, being a colorblind person, couldn't look at fly eyes. Uta gave me a chance in human genetics to clone Duchenne gene, but by the time I went there were others who already did the job. So I was devastated. She wanted me to work on cancer, and I said, no, I want to clone a disease gene. She was going to Stanford, so I moved to Shem Wiseman, who said, do whatever you want to do, as long as you are doing something good and doing something important. And that's where I sort of looked for genes that I want to clone. Next to Duchenne was gene for X-linked retinitis pigmentosa. I didn't know anything about the eye or vision. And then Neeraj Agrawal, who was at Yale and then had gone to Vision Field, then brought me to ARVO and introduced me to many doyens here, Gene Anderson, David Peppermaster, Holyfield, Matt Lavelle. God knows I had only known their names reading the papers. So thank you all for educating me in vision. Now, as a young professor, assistant professor, I went to Michigan. And my earliest mentor was, I would call Paul Lichter, who was our chair. And he never interfered in anything, never said anything. And he said, Anand, do whatever you can. Explore. And as a confused man, I tried to explore everything. I work on so many different things. And he supported me always. Another mentor is Paul Saving. Paul actually recruited me to help recruit me to Michigan. And he told me one day, he said, I want you to meet my patients. Paul, you remember that hopefully. He said, I want you to see the face behind the tube of DNA. And meet those people whose DNA you're working with, they depend on you. And that sort of was highly motivating, but not as much until I read this one. And I've cried so many times by reading this young girl this 6-year-old girl with LCA, Amy. I never met her. Maybe Irene or Steve Sang or somebody will remember who gave this to us. Her mom gave it to some of us who were talking. And she said, thank you for fixing my eyes. If you don't, I will still be happy, but please try. And that day I said, I have to try. I have to do something about it. So to that young girl who is blind, so I don't know how many times I've thought of her, thousands probably. So I work on emotional part aside, let's come back to science. I have been working on retina all my life, at least in vision since 1987. And in retina, I work on photoreceptors. I thought I will go to the real neurons because some people don't consider photoreceptors as neurons. They don't have action potential or whatever. But I've been stuck with those photoreceptors. And I've been looking at how these photoreceptors develop and what happens in aging and disease conditions in retinitis pigmentosa or age-related macular degeneration. So I'm going to tell you some very early stories, won't belabor, but I have to tell you, again, the mentors, clinicians, and friends who have, in my early Michigan days, I started with X-linked retinitis pigmentosa, and Paul Sieving, again, along with Jerry Fishman, and late Professor Sam Jacobson, God bless him, his soul, and John Hecken Lively. They were wonderful, wonderful colleagues, friends, mentors, clinicians, whatever. They helped me understand what retinitis pigmentosa is. We together published many papers. I got scooped by my own friend, Alan Wright, and who is very dear to me, and Shomi Bhattacharya. Both Alan and Shomi helped me tremendously learn about the disease as well as about life. Here is he's another very close friend, Gonzalo Becasis, who actually was not involved in X-linked RP, but AMD work. RPGR cloning, even though I didn't clone the gene, but we did quite a bit of work, led me to my love for primary cilia in those early days. And Friedhelm Hildebrandt was my colleague in genetics, who actually introduced me to some very basic concepts that the ciliary defects can be both important for kidney as well as vision. I somehow didn't really have the connection. He used to come to my office in Kellogg and come and tell me to work on these NPHPs, nephronesthesis, and I didn't care at that time until NPHP5 came. And then I was fortunate to clone independently CEP290 from mouse model, whereas Friedhelm's lab cloned CEP290 through Zobier syndrome. Along the way, many other disease genes we cloned that were involved in retinal degeneration, and I don't want to go through all that today. I must mention two people, Hemant Khanna, who is probably here, and he was very much instrumental in early days in the work on ciliopathies, as well as my colleague, Tiansen Lee, now at NEI. Now, I have to tell you, of course, NRL. During my early days, when I was trying to work on X-linked RP, I made a human retina cDNA library that was in 1987, 88. When the kits were not available, it was not that simple. I also developed the method for subtraction cDNA cloning, and one of the genes that turned out was NRL. That was probably the first retinal transcription factor or retina-specific transcription factor. Incidentally, PAX6, human PAX6 also came from one of my libraries, and NRL has become a big focus of our research over the last several years. But I'm going to just tell you very briefly about NRL. NRL, again, it was with Don Zak that we first showed that NRL regulates expression of rhodopsin. And actually, that's where our friendship began. We collaborated, but we also competed on some of the projects. So we have been friends and colleagues, and I truly cherish those wonderful memories for the last 30+ years. Alan Mears in the lab made a knockout of NRL and found something spectacular. The loss of NRL led to a retina with no rod photoreceptors. Instead, all photoreceptors, or most of the photoreceptors, were S-cone-like cells. And I remember Paul Sieving coming and telling me, I have seen RD mutants with flat ERG for rods, scotopic ERG, but S-cone, this photopic ERG was amazingly, so that was a sort of real, I feel, accomplishment that we had at that time together. And I have to say, Alan Mears showed that NRL is important to determine rod photoreceptor cell fate. Then another graduate student in the lab at O showed that if you express NRL ectopically in early cone precursor cells, they all become rod photoreceptors. Around the same time, Douglas Forrest was working on another transcription factor called TR beta 2, and he showed that if there's no thyroid hormone receptor beta 2, there are no M cones, instead there are S cones. Together, we gave this hypothesis, a very simple hypothesis, that NRL and TR beta 2 can generate all three type of photoreceptors in mouse retina, that is M cones, S cones, and rods. And NRL was dominant over TR beta 2. If you express both in the same cell, they all become rods. Masayuki Akimoto in the lab then took a couple of kb upstream of NRL and made a mouse with GFP expression and validated further the work of Matt Lavelle that rods are born as early as E12, and about 10% of rods are born before birth. And this beautiful sort of data, this work led to a lot of other studies later on, because this was the sort of foundation for later studies on idea or concept of photoreceptor transplantation. Hyunjin Yang and Jungwoon Kim, Matthew Brooks, and many others then worked together to look at rod gene profiles, integrated all this in Sarko's plot. So we now know everything what we need to know, at least gene-wise, not function-wise, when and how the rod photoreceptors develop. But you can also make these NRL-GFP mice with knockout mice and look at S-cone differentiation as well. Now, again, Don is again here because Ximing Chen and Don's lab identified this transcription factor along with Connie Sepko and Rod McInnes, this gene called CRX. And we showed that NRL and CRX synergistically activate expression of rhodopsin and many other genes. Ken Mitten in the lab directly showed that these two proteins interact, and he gave the sort of hypothesis of how this synergy may happen. And we are now working with Nick Artemaev and to look at the structural basis of this synergy by doing some of the crystal structure for the DNA and protein complexes. NRL is also highly post-translationally modified, but I'm not going to be talking about that story today at all. NRL mutations cause retinal disease. Almost all, actually, photoreceptor degenerative diseases are caused by targets of NRL and or CRX. So those are for some other day. Now, this is very important because NRL doesn't do its job alone. Along with CRX, there are many other transcription factor and binding proteins that are participating in this work. And it turns out many people in the lab showed that NRL interacts with a large number of RNA binding proteins. Jimena Corzo-Diaz, who just joined Stanford faculty last week, Jimena identified specifically proteins, RNA binding proteins that bind to R loops. Until 10 years ago, my students and fellows, they always teach me something new. Until a few years ago, I didn't know. I knew sort of, but I didn't know what R loops are. R loops are obligatory three-stranded structures that are made during transcription. These are DNA-RNA hybrids and display single-stranded DNA. When a gene is being transcribed, these structures are formed, and they have to be resolved. Their resolution is extremely important, and this is regulated by many different R-loop binding proteins, including these helicases like DDX5 and GHX9. And Jimena is going to be taking over that project, but the bottom line is that NRL not only is involved in transcription initiation, but it also is involved in elongation, transcription elongation process, which is very interesting. And probably it is participating in the splicing process. But that's, again, for some other day. I won't be touching upon those stories today, and let Jimena develop some parts of it. And I won't be talking anything about NRL after this. What I'm going to be telling you is a different story. And that sort of started with Shigeo Yoshida, who was in the lab in very early days in 2001. He published the first gene profile. In a way, I shouldn't say gene profile, but microarray analysis using slide microarrays. And in these slide microarrays, he looked at gene expression changes in aging human retina. And that paper we published, I think 2001 or 2, in IOBS, showing changes in expression of few different genes that were validated by RT-PCR. Since then, many things changed. Those slide microarrays were converted to Affymetrix microarrays, and now these NGS technologies or next-generation sequencing with not only RNA-seq, but many different kind of seqs, you can now look at all kinds of things. And this just shows the timeline. And I have had a great fortune to work with large number of wonderful fellows, Masayuki Akimoto, Jungmoon Kim, Hyunjun Yang, Ben Fadel, Laura Campeo, and Matthew Brooks, and Milton English. I just have few pictures here. I could have another 50 people here. I have been fortunate to have 500 plus students so far in my career. So thank you all. So what I'm going to talk to you in the next few minutes that I have are some of the key questions that we are now focusing on. First is, how is this genetic information decoded to generate specific morphology and function of retinal neurons, and particularly my favorite neurons, photoreceptors, both rods and cones, how genetic variations modulate healthy and diseased phenotypes in the retina, how aging and environment influence retinal gene expression and function, and how can we utilize this knowledge to develop novel paradigms for efficient gene-agnostic therapies. So this is a little different, not gene therapies, gene-agnostic therapies. So I'll be sort of taking the example of AMD to explain some of these things to you in the next few minutes. Now, age-related macular degeneration is personal to me, and my father had it, and I am homozygous for the so-called this big this gene HTRA-ARMS2 for that, supposed to be at risk, but Emily checks me every year, says so far so good. I'm 67, so I think I have a few more years. If I go with 20 years, that's the karma part at the end you are going to say. So AMD, it's not genetic. The most important risk factor actually is age. You have to be old to get AMD. That's what the name says. You can have all the genes, all the variants. When you are born, you will not get disease until you are 65, 70, 75 years old. And the third part of this triangle or trifecta is environment. So genes or genetic variations that you have, advanced age or aging, and environment. These are the three very important factors. It's multifactorial. It's not a single gene disease. It's very complex. And it's mediated by both genome and epigenome interactions. Genome-wide association studies like GWAS were initiated part of the consortia thanks to Heemin Chin, who was at that time in NEI, who initiated that. And the AMD consortium and later work by NEI, funded by NEI, showed that there are at least 34 loci, genetic loci, not genes. Loci is the region where these variants exist. So they are lead variants who are associated with disease. These lead variants, actually, a majority of them are in non-coding region. And we actually have no idea what those lead variants do, because not only the lead variant, but also other variants in linkage disequilibrium, they can be equally responsible for that. It's just that they are not seen for a variety of genetic regions. So genetics tells you about association, does not tell you the importance of any gene or pathway in this. So you have 34 loci, and some of these are real genes also associated with that, and that we know based on rare variants that are seen in those genes. If you have variant in a coding region, then you can say, yeah, that gene probably is involved. If it's in a non-coding region, how is that influencing the disease? Probably by looking at gene expression, by controlling gene expression. So a lot of people ask, so what does it mean by this, all these variants that you have got? Sudha Iyengar and her colleagues, Neil Pichi and many others, are now having 63 loci. And probably if you use 10,000 or 100,000 people, you may get another 100 loci. So there are many, many genes all over, many variants. How do they contribute to disease? So that's what we like to know. One way is that you look for rare variants or ultra-rare variants that are in the coding regions, because then it's easier to establish causality. And that's what we are doing. Thanks to my client and his enormous work over the last 40, 50 years, he has collected large families like this, and with age-related macular degeneration. And of course, Emily Chu, who has been our big collaborator in all these projects, Lena Zellinger and Jayashree Advani have been working with all the folks in this group. And we published one paper earlier showing ultra-rare variants in complement factor 8. But then Mike has got over 100 families that have been looked at by whole exome sequencing as well as whole genome sequencing. And we have been analyzing that data, and you will see some surprises. Probably we'll be submitting the paper in fall. There are many other interesting signaling and other pathways very strongly sort of showing up based on based on rare variants in the large families. Now, coming back to non-coding variants, how do those non-coding variants affect gene regulation? You can have a variant here in this region. It can regulate expression of the next door gene, or it can regulate expression of gene maybe hundreds of kilobase pairs away. How does that happen? We were very fortunate to work with Deb Farrington, who's right there, smiling. And I was just walking with her one day, and I asked her, oh, it's so hard to get retinal samples. And she said, how many do you want? I said, I knew 80 was a GTEx thing. I said, maybe 100, 150. And she went back to her freezer and said, will 200, 300 be enough? And I said, yes. Then a week later, she said, I have 200, 300 more. And then she goes back, and now we have over 1,000 samples from her, from peripheral retina, macula, pigment epithelium. It's great to have good friends, and great to talk at conferences, because you never know what you will do next. So Rinki Ratnapriya, who is now at Baylor, Rinki took up on the challenge, worked with a large group of individuals, analyzed 500 post-mortem retina samples, and showed by genotyping and transcriptome analysis that over 10,000 genes are under genetic regulation. And people were saying, why are you doing this in retina? Why not take from other tissues? She showed clearly that at least 40% of those EQTLs, that's what we call them, they are retina-specific. So that shows the value, and that sort of helped us put together the candidate genes at least at 6 genetic loci, because those non-coding variants can regulate expression of any number of genes, hundreds of kb away even. This narrows down to few candidate genes. But that's one part. These SNPs, or what we call a variant, can also regulate methylation. And those are called MQTLs. And some of these methylation sites can regulate gene expression as well. Those are called EQTMs. Why are they important? Because aging and environment do not directly impact your DNA. They impact your epigenome. And the most stable epigenome mark is DNA methylation. So working with Bernard Weber and my good friend Ayelet Segre, who actually teaches statistical genetics to me all the time, every couple of weeks when we meet, and Jayashree Advani, an amazing postdoc in the lab, they work together. I just take the credit. So I just talk to them every time. I just put my 2 cents, how is that, why to do this? What they did was they did the QTL mapping of whole DNA methylome. And that methylome analysis identified at least 87 gene epigenome interactions. And what came out? Came out were these pathways, including metabolism, glutathione, glycolysis, the pathways that never came by jivas, but were always shown by biochemistry and other cell biology studies that oxidative stress is important, mitochondria are important, metabolism important. But none of the jivas loci was showing that. This came from epigenome. That makes much, much better sense. And that's thanks to, again, Ayelet, Jayashree, and many others, Milton. So now, again, coming back, how are those variants regulating gene expression? Now, these variants, just think about the DNA. It's a 2-meter-long DNA strand that is packed, condensed within a 5-micron diameter nucleus. That condensation of that DNA is not random. That topology is very unique to each cell type, and that's what you would like to know, because that genome topology is dynamic. It changes with differentiation, with aging, with time, and in different cell types, it's going to be unique because it has to facilitate expression of different sets of genes. And these variants, if they're far away, they make these chromatin loops. And that looping brings those regulatory elements close to the promoter of appropriate genes. And that chromatin looping and this organization can be elucidated by a method called Hi-C. You can use that by genome-wide mapping. And that's what, again, I didn't do anything. Claire, Nivedita, Jayashri, Zach, Jimena, and Kathy, what they did was they did enormous amount of work. They did retinal Hi-C. They obtained that Hi-C data, showed there are 60,000 loops. They integrated that by, with epigenome marks that we created some, and some by beautiful work done by Tim Cherry as his colleagues. And what they did was, by integrating these, they identified cis-regulatory elements, enhancers, super enhancers, and integrated all of that data with GWAS from both AMD and glaucoma, and that gave us a lot of insights. And I'm going to give you one example. This is the locus by GWAS called C20ORF85, I believe, if I remember correctly. And this is the variant, and these are the variants underneath in linkage disequilibrium. So theoretically, and this is the nearest gene is C20ORF85, so it was called that locus. But when you do Hi-C, you see this region is making loops somewhere here in the left, and you have other genes that are in that region. So that means these are likely candidate genes for these variants and not the ones which are next door. So what this Hi-C study is doing is it's going to refine the areas and give you candidate genes for AMD, what real targets are. And this is about 300 kb away, not next door. And this is true for almost all loci. And this is true for glaucoma. You can utilize that. We have done that. Zupeng Q has done that in organoids of human retina. This can be done for developmental diseases as well. It happens in aging too. This genome topology can change in aging, and consequently it changes gene expression. Again, Claire, Nivedita, and Jimena, and others have been working on this, and Matthew. So what they are showing is that this is using the mouse aging data, and this is, again, some very new stuff which we are showing that in aging, the topology is changing, and consequent gene expression is changing. Now, I'm going to come to the last few slides, science slides, and going to tell you my interest in gene agnostic approaches. In 2002, in one of the meetings, I suggested that we probably need to look at, there are going to be many, many genes associated with retinal degeneration, and the common pathway has to be there because these genetic mutations have to converge somewhere because there are only few limited number of pathways that lead to cell death, photoreceptor cell death. In all of these diseases, ultimately photoreceptors die. So how is that happening? Again, as I said, the genes are part of networks, and I showed you a few of those. And these networks, if you disrupt a primary node or hub, you can disrupt the whole network. If it's a secondary hub somewhere in the corner, it doesn't have much impact. But if you know the knowledge, if you have the knowledge of these networks, you can try to tweak them. You don't have to actually change this hub back again, or you don't have to do anything. You can tweak this and make it into equilibrium once again. So the idea is if you define commonalities in degeneration pathways, then those pathways can be targeted. And it turns out that some of those pathways, again, relate to mitochondria, a field that I left some 40 years ago from my PhD days to clone the genes, coming back to mitochondria, energy homeostasis, autophagy, proteostasis, thanks to Alan Taylor, who is teaching me every time what happens in proteostasis. So you can look at those pathways and try to tweak those pathways to develop drugs. Now, my Polish brother from a different mother, Chris, probably doesn't recognize that, but several years ago when we were collaborating, I realized that pharmacology could be a way to do this. And Holly Chen in the lab worked with my wife, who works, Manju, who works at NCATS, and this idea came during our sort of dinner conversation one day to do the high throughput screening at NCATS, and Holly really very elegantly worked and discovered, after screening 6,000 different compounds, 5 compounds, they all worked very well. Here's one compound, reserpine. You can see how reserpine can protect these cells in disease organoids. These are from CEP290 LCA patients. They can keep the photoreceptors alive. Since then, we have looked at many other compounds. You can see A3, C2, and F2 here, which work even better than reserpine here. And this is in P23H female rats. And this is, again, a collaboration with ICRO. And these compounds seem to also work in RD10 mice. So in 3 different animal models and also in organoids, some of these compounds seem to work. So you can actually target pathways and design drugs which work with more than one disease. And we think we are getting somewhere. And hopefully, in the next few years, maybe the promises that we have made to, I myself made to Amy and some others, maybe we'll be able to do something about it. Now, here's the karma. I showed you this. Genetics, aging, and diet, which Emily talked about, lifestyle and environment. In our Indian culture, we call it bhagya, kal, time never stops, and karma is really your diet, lifestyle, environment. Karma is actually your actions. It's not good or bad. Karma is karma. So there are 3 things: genetics, aging, environment, bhagya, kal, and karma. You can't change that. Your time is not going to stop for you. You have the genes that you have. The thing that you can do is, as Emily said, diet, karma, your actions, your lifestyle. That's easier. Prevention is better than cure sometimes. While in the meantime, we do try to change aging, while we try to change our genes, I think easier thing is our karma, that we can change that. We can change our epigenome probably, and change the trajectory of the phenotype. This is not going to change what is going to happen, but it can change the severity of disease, and also it can change the onset of disease. So that's where we are. I'm going to go back to my colleagues from University of Michigan, and this is how it was in '92 in my early days when I did have some hair. This was many of my colleagues here. You can see by the time of leaving Michigan, this is in 2005, my lab was fairly big. This is in NEI. These are parties at our home. These are lab in those days. You know, this is how it used to be. This is how now, the parties in the lab, you see all these women power. I mean, they, you know, and you can see how all that is. So you have a lot of fun, but you work hard and play hard. That's the way it is. My collaborators, I think there are hundreds of them. I wanted to show everybody's picture, but I cannot. And I think I may have missed 1 or 2 here, but these are all the people I've published with. And there are many more who I have worked with, and they get all the credit. I really, again, I don't know how I did it, but there are just so many of them, wonderful people. So thank you at University of Michigan. When I was NEI extramural program, Lorianne, I have to shout out for her, Himin, Grace, many others. They are in extramural Foundation Fighting Blindness, RPB, Macular Vision Research Foundation at NEI. internal program. My scientific director, Sheldon Miller, who recruited me, he and I sort of used to have fun with RP versus photoreceptors. I always told him that photoreceptors are the king and RP is the queen, like in chess game, you know. But you know, if the king cannot live for long if the queen is dead. So, but you still don't lose the game if the king is there. Anyway, and I have to say, David Schneeweiss has been wonderful. And now we have our new scientific director, Kapil. I hope he keeps me there for some more time. And Mike Xiang, our director, they're wonderful, brought a lot of energy. I have to say 3 of my colleagues, the co-directors, who have been very instrumental in some of the work that we do, Bob Ferris, Hao Kwan, and also Li Jindong. I've had 3 assistants in my whole 35 years. Dorothy Gable was the first one. I couldn't find her picture. She was with me last time in '97. Sharon has been with me and taking care of all the work that I do for a long time. Sharon Ferrara and now Lucia. But Lawrence, and I have to shout out, without them, my lab will not run. And of course, my family and friends. This is my grandson, my granddaughter, my 2 kids. And this is my family, my in-laws, they're all here. And there are many friends that I have made personally, and it's amazing. Now, this is our family, and my kids keep me grounded, whereas my grandkids let me fly. They tell me what is important in life. They show me every day, and I'm so thankful. And this is sort of our daughter, son-in-law's dog, Albus, who we take care. And of course, this is the love of my life. I met her 42 years ago. I don't know, after 2 years, 2 hours talking to her, and she said, you were the only one talking, and you can believe it, that is true. She still agreed to marry me. I gave her 24 hours to decide, and she still did that. And she's still with me. And she tells me what is right, what is wrong. She's my worst critique and my best supporter. She has been with me in thick and thin. And I can't say enough about her, but she has become my best friend now. More than my wife, she's my best friend. I have a long way to go. This is not the end. This is still, the journey continues. I have promises to keep to Amy and to many other kids. And this is my more recent, I love to teach, and as my daughter said, or my son said, whoever said that, Tamso ma jyotirgamaya means take me from darkness to light. And I'm trying to do my little bit, taking some of the kids to their light, whatever, doing my part. So thank you very much for listening.